Hi, my name is Bob Rosani with the FSHD Patient Advocacy Group. Thanks for joining us again. We're on our third visit as we tour the North America continent, the first continent. We've been at two facilities. You've already seen Peter Jones' lab as well as Scott Harper's that are doing molecular biology for treatments and or potentially cures. And I have the pleasure now of being in our third facility in University of Kansas?
Yeah,
or Kansas
University of Kansas Medical Center. That's
Thank
right.
you.
Yeah.
He'll correct me. So Dr. Statland, Jeff Statland here and his group also work on research, but not from a molecular biology type of perspective, but more from clinical measurements, clinical outcomes, things to support potentially trials that take place and give good data sets as to the efficacy of any potential treatment that may be coming online. So with all that, I'm going to turn this over to Jeff, and hopefully we have a little bit of a dialogue. So,
Sure. Yeah. So I think that my training is really in the design and conduct of clinical trials in people. Like you already said, we call it experimental therapeutics. But everything I do is a collaboration. So maybe a little different. I mean, everyone collaborates, but for me to do my work, I have to work with all the people you mentioned. Peter Jones, Scott Harper, and what we're trying to do is to help them take their innovations and create the pathway, the infrastructure, and the tools so they can then go and test them in people, because we need all of these pieces working together to get effective treatments for FSHD. Yeah, and so I started working with Rabi Tawil. He's at the University of Rochester. He's my mentor, and he trained me. But we started working together in 2010. At that time, they had just published the unified model for FSHD, which identified a molecular target for therapy.
Mm-hmm.
And we realized at that time we really needed to start getting in gear so we'd have the clinical tools that we would need so we could measure a response to therapy in the setting of a clinical trial. Because we were all very excited that this was setting the stage for these molecular-based therapies. And so we took a very systematic approach. We started with what tools were already there, what clinical trials had already been done, and a number of them had been completed. There was an albuterol study that was done in the US for FSHD, and then there was the Wyeth myostatin study that was done. And so we pooled all that data together, and we said, well, this is what we have. We have a pretty strong start. We had also had a natural history study in the late 90s, and so we knew a lot about strength and about what happened to people in time frames of about a year, but there were still several gaps that were missing. We then went and we organized a number of international workshops. So Rabi Tawil really took the lead in this with his colleague Sylvain Vandermaal. And we had two large workshops with the FDA, various researchers, clinical physicians from across the world who came together. And we said, well, we still need a lot of things. We need to have a better understanding of what are those things about someone that we can see when we first see them. The genetics, gender, age, how much you can or can't do that might better predict what's going to happen over the next year. You're probably aware of this. A lot of people always tell me, it depends who I'm talking to, but I'll hear things like, well, I feel like, I'm doing pretty good, and then a muscle's affected, and I lose it really quickly, and then I'm doing okay. And then other people will tell me, no, I think it's just been slow, steady change over time. So there were things we didn't really understand about how things progressed, and what are those things that will help us predict who's going to be who. And so we sort of systematically went into categories. We said, well, we're going to need things on the level of strength, on function, things that are patient reported, how you feel about it. It's very important, I think, to understand that perspective.
Yes.
But also biomarkers, so things we can measure in tissues. And we really took that whole thing, we said together we're going to call that a clinical trial toolbox. And so a lot of my work has been to create that clinical trial toolbox for FSHD.
Right.
Yeah.
Can I share some of the experiences that I've had from the research perspective?
Yeah, sure.
Okay. I came across Peter Jones, we did on YouTube. And the one thing that struck me, he wasn't even aware, he tells me, that video was made when he was doing a presentation.
Yeah.
But if you look at the thumbnail, his hands are up like this.
Yeah.
And I said, this is a guy who's passionate about the work. And it just so happens, I was in the East Coast, I'm now out in the Phoenix area. we had a show in Las Vegas, I felt this was a perfect opportunity to go up and see Peter and Takako Jones's lab.
Yeah.
And he took us through a whole bunch of things, including looking at the DUX4 mouse model that he's been working on, or Takako produced. And we were looking at slide cross sections and things like that. And I think, to go to one of the points you were just speaking of, their perspective may be, you talk about this, is it a ladder? You plateau and then you drop, you plateau and you drop. And Dr. Laura Ellman, who works not so much specifically with FSH, but she was a part of University of Pennsylvania.
Yeah.
She was an early ACE83 Acceleron trial, which I participated in one very small cohort of the open label, for that matter. And but Peter feels perhaps, or that what's happening is, let's say using my quads, where I'm really getting affected now. That if obviously the muscle is consisted of a lot of myofiber or whatever strands, like a rope. And is it possible that they believe, or they're feeling, maybe you lose a little bit of strand at a time, and you're not really aware that you're actually progressing until you reach this inflection point or critical mass of enough diseased or dead muscle tissue that you feel it. Yeah. So, is that possible? I mean, the disease, as you know, presents itself extremely variable. So I can only speak from my own particular experiences on my journey on how it's been impacting me as well. So hopefully that'll be some interesting.
Yeah, well, so there's a lot there. So the Acceleron study, you're right, we have a clinical trial in FSHD going on. So I don't know if you knew this, so I'm the lead PI for that study.
Oh, no, yeah. I'll shake your hand for that. Thank you.
Yeah, and the
ACE83, beautiful.
Yeah, but it's Acceleron study, and so in full disclosures, I'm a consultant for them. So not my study in that sense, but the I think that's the key question is right, how is it progressing? And it may not be the same for everyone, but I think that we're sort of evolving in this concept, because you're right. What I used to tell people was, well, what's likely going on are your muscles are affected, right? And you're having this slow loss of a few fibers at a time, and you're not going to notice it because the muscles are fairly large, but
right.
You're going to reach that inflection point where your actual functions altered, and then you're really going to notice it. For example, the shoulders, you're going to reach a point in time where you can no longer reach over your head,
right? And
that's a big change, right?
Yes.
And exactly, or you're going to reach the point where that knee doesn't lock anymore, and that's going to be a big problem, because that's when the knee starts to buckle. But I think more recently there's been at least a little bit of evidence that there is some signal you can see, and they've really done this more using MRI studies.
Mm-hmm.
Where it appears that, for whatever reason in FSHD, we can see some signs on the MRI of what would be inflammation, so fluid in the muscle. And you see this in all dystrophies, but for FSHD in particular, this sort of correlates to, if you take a biopsy of that muscle, you can see some inflammation in the muscle, but then we can also see these molecular activity related to the expression of DUX4. And so it may be that there are some signals of this activity. Now, what that means for someone's experience of it, I don't think we know that yet.
Right.
But it may be a tool for us to screen muscles that are at risk for changing over a shorter time period, or muscles to target in an early therapy. I think when I'm thinking about, what can we do to start addressing the research enterprise and to sort of reach out to people to get them involved. So everything I do is a collaboration. I'm not sure if you're aware of it or not. So in 2016 with Rabi Tawil, my colleague, we put together a clinical trial research network, and so for FSHD. And we did this because at the time we felt that we were hearing that there was this work that needed to be done on a clinical side, and nothing was really happening. It wasn't getting done. Every year I'd hear the same thing. Well, we really need to do these studies, and it wasn't being done. So we just finally decided, and it was very informal initially, we're just going to get some sites together to collaborate to answer these questions. And so Catherine Wagner was one of those original people, Rabi Tawil, myself, John Kissel, he was still practicing then at Ohio. But the network's grown pretty quickly since then. So now we have 8 sites in the United States that are in the network. We have 3 collaborating sites in Europe that are participating. We have a number of studies involved. We have the largest study in the U.S. that's ever been performed. I think you may be in it. That's the RESOLVE study.
Oh, yes,
Yeah,
indeed.
right. And so that was the study we put together to run on this network. And
Wow.
and it's really an important study, a study like that, because you're going to help us answer those questions about the different things that can predict progression, as well as validate some of these tools, the ways we're going to measure things. But the heart of the network, what we did is we said, it has to be a collaboration. And so we've been involving people with FSHD from the beginning. So they're on the advisory council for our network. We worked with the FSH Society initially to set it up.
Sure.
We have several individuals who gave money initially to help with the network. And then we have what we call advisory councils, so people who either are in focus groups or helping us out, and they help us run it. So all things related to recruitment, how we do the studies, and this is something we've heard from people, which was people don't have a good understanding of what it means to be in a study. And so we're going to try to, Kylie here is going to try to help.
She's off camera at the moment. But
yeah, she's going to try to help put together some materials to help people with this, or we were thinking of creating some videos as well to help answer this.
Great.
And I know, like here, when people come here, I always think, this is a it's a tremendous thing when you go volunteer for a study. I mean, it's people who volunteer for the studies that help us, make any advancements that we make in understanding
Sure.
disease. It was people volunteering that helped us answer that question about the genetics. It was the large families that had volunteered at Rochester at that time, and then also in the Netherlands. And it's people volunteering that are helping us with the clinical questions. So when someone comes in here, I want it to be a good experience.
Sure,
So, I think they come in, and I always try to talk to them so they understand what we're doing, why we're doing it. I try to make a real strong effort to get the results of what we do back to people. So if you're in a study, you should always ask for the results when they come out.
If you see these videos or you're in Facebook, please join. And we need that data. The quicker we can get these sampling groups, the better your data results are. It's going to help short circuit all the effort that here Dr. Statland is working with all the others. So please participate.
Yeah, I think people, it's time to be involved for a lot of reasons, for that reason, but also because, we are looking at the reality of companies coming in and doing these clinical trials. And so I think, as a community, we have to start asking ourselves, what would a treatment mean? What should it look like, right? Who's it going to be for? How do we figure this out? What's going to be a meaningful change? And so, I think that we're going to see more and more of it. There's one company doing a study now. There was another company doing a study a few years ago, ATIRE. I think there's some companies who have studies planned really in the next year or two, like Fulcrum, who's already got a trial preparedness study of their own going on right now. I think there's a lot of companies, and there's people like you spoke to, Scott Harper, who have their own, what their labs are creating, that I'm hoping we'll start seeing those being tested, in the next, say, 1 to 3 years maybe.
Right.
And
Yeah, and in fact, I think they said when I was there, we may be working on some of your muscle when they did the xenografting. So I have real skin in the game, or muscle in the game.
Yeah, you have real muscle. Yeah, and if you ever, I always tell this, so people who've been up to Rochester, their muscle goes a long way,
right?
Because they immortalize it into these myoblasts, and they give them out to all these labs.
Yeah.
So it's really amazing. Just that little bit of participation, it has an amazing impact.
Right, indeed.
Yeah.
One thing I'd like to talk about from an experience is I have no patella responses anymore.
Okay.
And that's gone downhill over the disease progression in the last 6 years. So you can hit, as Daniel says, the same thing. You can hit his knee with a ball peen hammer, and he gets no response.
Right.
And
I'm asking, yeah.
Okay.
okay, and I think Scott's group said he feels that anywhere DUX4 is expressed, there's apoptosis going on. So whether it's in the muscle tissue or potentially other things, it leads to that. And one of the things that I feel as an experience that I'll share with you is as I've gone downhill, there's quick twitch muscles, there's slow twitch muscles, right? And in that, there's this interface between the nerves, going into the muscle. There's that bridge that signals that. And my belief is that if DUX4 is expressing in the muscle, and I know it's hard to see it wouldn't surprise me that it's going into that interface between nerves and that, because I see the connection. As I weaken, I feel that I can't get the response. And so is there a neural, even if I'm doing velocity conduction tests that don't show anything unusual necessarily, is it possible at those areas where there's pathology going on specifically, is there a component that is leading to nerve damage? It's just a feeling that I have.
Well, so I don't have an easy answer for you. What I would say is that relationship between the nerve and the muscle is a two-way relationship. If you affect the nerve, you affect the muscle, but if you affect the muscle, you're likely affecting the nerve on the presynaptic side. And so it's certainly possible there's changes going on at that junction, and I don't know that I have the answer for you, though.
Yeah, and when I presented that to Scott,
that would be something to look at, though.
Yeah, and I think when I brought that up to the Scott's group, they thought that was a very intriguing idea. So maybe something good comes, from that, obviously. And so far as, again, about the ACE83, I'm not going to prejudge it. I'm just going to tell you my experiences as an individual. I think, I was shot in my tibialis anterior, so I didn't have my uppers. And I did believe I got some increased mass. The question is, as I'm sure you know, the burning question now is that usable muscle? Or is it a bigger, generating in the context of, do you really get production out of it? So another thing I suggested to Scott, and why it's so important, I think, for collaboration, is it wouldn't surprise me that you have a regenerative therapy also with knocking down DUX4. So can you bail the water out of the boat quicker than the water's coming in conjunction with, together as a dual bifurcated approach to treatment, as opposed to just knocking DUX4 down?
I think, I can't speak for the company. I think the company they have presented results. So I mean, in the part 1 where they were just trying different amounts in the
doses,
muscle,
right?
They saw increases in the volume. So I think your experience was what they saw. The bigger question about whether that's going to equate to strength or functional benefits, well, that's why they're doing the study now with the placebo-controlled trial.
Right.
To get an answer from that. But I think your idea that a drug like that would be used in conjunction with a disease-modifying therapy is appealing to a lot of people. The idea would be that, well, we'll use a drug that maybe would stop the molecular mechanism of DUX4. In the meantime, though, we want to have some sort of restorative drug that could really help someone maximize the benefit. And it may be down the road there would be a combination. Just from the regulatory side, it's a little more complicated because they'd have to be approved, or a company would have to be ready to try those in combination. But I think it's a good idea. Yeah.
I think you did fantastic, and again, thank you. I hope we were productive in our conversation.
Well, no, welcome. I'm glad you came. What do they have? I think there's some lunch here coming for you. And
we're in Kansas City, so they got ribs or barbecue, right?
Yeah, it's barbecue for Kansas City, and you can meet. We have our whole research team. I think they're around here, and you'll start to see them pop up once there's food, probably.
And if you ever had any,
and I'll be
questions
around.
for me, I'll be happy. I mean, if there's anything, and I'd also offer if you have any preclinical type stuff you'd like me to participate. I've already been a guinea pig at both facilities.
I tell you what, we have studies now. You're doing the Resolve study. You're doing it in Baltimore, or?
Yes, sir.
Yeah, right. And so you might want to do it there because she's also got this. So we do have other studies going on now. The MRI and the molecular biomarker studies, I think, are particularly important. Most of the companies who are thinking about these disease-modifying therapies, they think that the way they're going to tell the drugs are working is by looking inside a muscle before and after they give their treatment. In order to prepare for them to do that, we have to know what they're going to see in that muscle.
Understood.
Yeah, and so we have these studies going on now to really address this question, that relationship between what you see on the MRI, what's in the muscle, and what happens molecularly. And so I think this is an important question.